Previously, we covered the fundamentals of light and fluorescence spectrum overlap. Today, based on spectral overlap and the detection principles of flow cytometers, we'll discuss fluorescence compensation and the characteristics of common fluorochromes. Fluorescence spectra often overlap. For example, with 488 nanometers excitation light, there is overlap between the emissions of FITC and PE fluorochromes. The emission light of FITC partially overlapped with PE. Flow cytometers detect fluorescence within the scope of a certain wavelength of light signals. For instance, a 530/30 filter allows detection of light centered at 530 nanometers with a bandwidth of plus or minus 15 nanometers, thus suitable for detecting FITC. PE, on the other hand, is typically detected using a 585/42 filter. From the image, you can see that there is some emission light of PE leaking into the FITC detection channel, while a significant amount of emission light of FITC spills over into the PE detection channel. How do we adjust compensation? When performing experiments, if there's spillover between fluorochromes, we need to set single positive controls to measure how much of each fluorochrome signal spills into adjacent channels. We then subtract this spillover during data analysis, aligning the cell populations horizontally and vertically in the plots. Besides considering spectra, the intensity of fluorochromes is also crucial in panel design. Different cells express varying levels of proteins. Some are highly expressed, while others are low. When designing panels, we need to follow the principle of matching bright fluorochromes with low-expression markers and dim fluorochromes with high-expression markers. You might wonder, how do we determine the brightness of a fluorochrome? Fluorochrome brightness is indicated by the staining index. This image shows the SI values of common fluorochromes. As we can see, PE and APC are very bright, while FITC and PRCP are much dimmer. Additionally, understanding the properties of fluorochromes is essential for choosing the right ones. For intracellular or nuclear factor detection, where fixation and permeabilization are required, many tandem dyes are unsuitable due to their instability. FITC, for instance, is unstable in acidic environments. For such cases, Elabfluor 488 can be used as a substitute. That wraps up today's flow cytometry class. Follow us on YouTube by scanning the QR code or find us on TikTok by searching Elabscience. Let's unlock more flow cytometry knowledge together.